Welcome to this explainer. We're diving straight into the computational magic behind bioinformatics today. Have you ever stopped to wonder how scientists actually read, interpret, and compare the fundamental blueprints of life? Well, that's exactly what we're getting into. We're unpacking sequence alignment, which is honestly the core technique making modern genomics even possible.
Here's our quick roadmap for today. We'll cover the language of life, how sequence alignment actually works, the rules of the game, the algorithms and tools we use, why this all matters in the real world, and finally, a glimpse into the future of genomics.
Let's kick things off with Section 1, the language of life.
Think of sequence alignment as the ultimate biological spell check. It's the computational method of taking two or more biological sequences and lining them up so that similar or identical characters sit right next to each other. By maximizing that similarity, researchers can spot shared evolutionary ancestry, structural relationships, or pinpoint exact functional characteristics. It's literally how we figure out what has stayed the exact same over millions of years of evolution and what's mutated.
Now, the specific language we compare depends entirely on what we're trying to figure out. On one hand, you've got DNA and RNA sequences, comparing those familiar nucleotide bases, A, T, G, and C, or mRNA, rRNA, and tRNA. You'd use this when you're hunting down gene mutations or doing comparative genomics. On the other hand, you have protein sequence alignment, where we're comparing these massive, complex chains of amino acids. And that is absolutely vital if you're trying to predict a protein's function or map out its three-dimensional structure.
Moving right along to Section 2, how sequence alignment works.
When we actually compare these massive strings of biological text, we usually break our approach into two main categories based on scale. First up is pairwise sequence alignment. This is when you're comparing exactly two sequences. Super useful if you're analyzing, say, a single mutated gene against a healthy one. But when we need to zoom out for a broader view, we use multiple sequence alignment, or MSA, which compares three or more sequences simultaneously. That's the go-to method for tracking evolutionary relationships across an entire family of proteins or building out complex phylogenetic trees.
Beyond just how many sequences we're looking at, we have to decide how we want to compare them. And that brings us to global versus local alignment. You can think of global alignment as comparing two books cover to cover. It forces an alignment across the entire length of highly similar sequences, right from the beginning to the very end. Local alignment, though, doesn't force that end-to-end match. Instead, it scans for the most similar partial regions, domains, or motifs. It's kind of like searching a massive library just to find one specific famous quote. It's absolutely perfect for sequences that might only share a tiny but biologically crucial fragment of similarity.
Let's head into Section 3, the rules of the game.
These algorithms aren't just taking wild guesses. They're playing a highly mathematical, points-based game to evaluate similarity. Step 1 is the match. Finding identical residues gets you a positive score. Step 2 is the mismatch. When the characters are different, it implies a potential mutation, so you get slapped with a negative score. But step 3 is where the real magic is, the gap penalty. See, in biology, genetic material gets inserted or deleted all the time. To align these sequences, the algorithm sometimes has to introduce empty spaces, or gaps. But if those gaps were free, the computer would just wildly scatter letters everywhere to force a perfect match. The gap penalty discourages that nonsense, ensuring the final alignment actually makes biological sense.
That brings us to Section 4, algorithms and bioinformatics tools.
Bioinformaticians have a pretty robust menu of tools to choose from, depending on the job at hand. For global alignment, the gold standard for dynamic programming is Needleman-Wunsch. If you're trying to find hidden motifs using local alignment, you'd pull up Smith-Waterman. When you're searching against massive databases, BLAST is your best friend because it uses heuristic methods for incredibly fast similarity checks. And for multiple sequence alignments, you've got Clustral Omega for standard comparisons, Muscle for rapid high accuracy, and MAFT, which is perfectly designed to handle huge, large-scale genomic datasets.
A couple of other tools definitely deserve a quick spotlight here. FASTA is a lightning-fast sequence comparison algorithm that gets a lot of use for database searching. And when a researcher needs an absolutely premium top-tier alignment, they turn to T-Coffee. It acts kind of like a funnel, uniquely combining results from multiple different alignment methods to distill one highly accurate consensus. Together, all these tools give researchers an incredible computational tool belt.
All right, Section 5, why sequence alignment matters.
This computational math isn't just theoretical stuff living in a server farm. It directly leads to life-changing, real-world applications. In genomics, it drives gene discovery and helps us annotate genomes. In evolutionary biology, it's how we construct phylogenetic trees, mapping the whole history of species. Over in medicine, it's literally the backbone of personalized medicine and pinpointing specific disease-causing mutations. It's essential for synthetic biology and biotech. And right now, in drug discovery, sequence alignment is exactly how we rapidly identify drug targets and develop new vaccines.
Of course, it's not a flawless system. We've got to balance its sheer power with its limitations. The advantages are pretty undeniable: detecting crucial mutations, identifying conserved genes, and really deepening our understanding of biological processes. But the hurdles are significant. For one, the tech is insanely computationally demanding for large data sets. On top of that, accuracy drops off a cliff when you're comparing highly divergent sequences, organisms that are very distantly related. And finally, as we mentioned earlier, a poorly placed gap penalty can completely distort the biological interpretation of your data.
Let's wrap up with Section 6, the future of genomics.
This field is evolving at an absolute breakneck speed. We're already seeing massive leaps thanks to high-performance computing, cloud-based alignment platforms, and the integration of multi-omics data. But looking ahead, the integration of AI-assisted sequence analysis is pushing us towards something that sounds like science fiction: real-time genomic sequence alignment. We're entering a world where large-scale genome comparisons happen almost instantaneously.
To sum it all up, sequence alignment is a core bioinformatics technique used to compare DNA, RNA, or protein sequences, revealing similarities, evolutionary relationships, and functional information that are essential for genomics, biotechnology, and biomedical research. It really is the fundamental bridge between raw biological data and actionable scientific knowledge.
So as you think about these rapid, mind-blowing advancements we just covered, I want to leave you pondering this. As AI continues to optimize these algorithms, slashing the computational load while boosting accuracy, what breakthroughs will we discover when we can instantly align and interpret every individual's genome in real time? How is that going to reshape personalized medicine, our understanding of evolution, and the very future of our digitized biology? Thanks so much for tuning into this explainer, and keep asking those big questions.